So now I'm happy to introduce the next, and that's Christoph Gerber. He's a Swiss professor of physics. He has been at the University of Basel since 2004, and he's a director of scientific communication at the National Center of Competence for Nanoscience. After his PhD, Gerber moved to Sweden, and in 1964 became a group leader in research and development for the company Contraus. In 1966, he moved to IBM Research in Zurich, with which he remained associated until 2004. In the 1980s, he also worked temporarily at IBM Almaden and at the IBM Physics Group in Munich. In the early 1980s, he worked with Gerd Binnig, Heinrich Roeder, and Edmund Weibel on the developments of the scanning tunneling microscope. He then continued his collaboration with Binnig, and while at IBM Almaden, the two scientists, in collaboration with Quate, realized atomic force microscope. In the last 30 years, he has continued to explore the possibility of using scanning probes as imaging, manipulation, and diagnostics tools. He is particularly interested in developing biochemical sensors based on atomic force microscopy. Please.
Thank you for this kind words. It's a pleasure, a privilege, and a tremendous honor to be here. So it was, as Gerd already alluded to, it was a fantastic time we had it way back together with Carl's great group. And as Gerd said, you know, some of this highly innovative people are with us today. And I would like to actually give you a sort of a impression the way we have taken AFM technology way beyond imaging and into the realm of personalized diagnostic and preventive medicine. And Gerd already has taken all the punchlines, so I better stick to the science, okay? So there was a lot more, but we don't talk about that. Anyhow, so there are about 200 different kinds of cancers identified so far. And most of them actually are driven by abnormalities in genes. And it actually requires many more mutations to be identified in order to develop the right strategy for the right patient. And of course, this requires also an early and fast diagnostic tools. And just to say that in the US, this year, a new program has been launched, supported exactly for this, supported by $1 billion. So this is what we are doing, we use batch microfabricated arrays of these cantilevers you just seen, and we made arrays of it. But there are no tips. There are no tips here, as you can see here. There are no tips. We just use the surface as a biosensor, and we apply that in personalized healthcare. So this is the operational mode. As you can see, molecules are functionalized on these arrays, and due to the induced stress between the molecules, the cantilevers or sensors are bending, and that is not the weight. It is the induced strain of the molecules that basically bend the sensor. And as you can see here, we are using references, and this is very important because if you don't do that with this system, it would be absolutely meaningless. The results would not really lead to a sort of result. And here we can do that in using actually as a diving board, but We want to keep the molecules on board, not like in the Olympics where they jump off, you know. And you can see what you can do here is this gives you a mass or a mass change, and the static mode, what we see here actually gives you a conformational change of the molecules, and you can do that simultaneously. So this is what we actually are doing, and this is transduction, converting biochemical processes into nanomechanical motion. That is what we're doing. And we do that in a differential measurement. I alluded that before because of the references, and that is taking care of all the side effects like temperature drifts and non-specific bindings and so on and so forth. It's an instant calibration, and the response time is actually in milliseconds. And that is how it is done. It's self-assembled, single strands DNA or RNA, and we do that with inkjet technologies, inkjet spotting, as you can see that, and then up on hybridization. The complement is squeezing in and creates that strain, and it leads to either a compressive or tensile stress, and is a mostly steric hindrance. And we also know exactly how many molecules we are actually looking at by we've done that in the hybridization efficiency, is what was verified using radioactive labeling. And to say the way it is binding here, it's a short alkane strand and with a sulfur at the end, and that does the binding, covalent binding to the gold surface of our device. So this is what we have done so far. We looked into mutational analysis, monitoring transcription factors, monitoring transcriptional activity, and analyzing protein synthesis. And this is some of the literature, what we have published so far. So this is actually what biology requires. In terms of sensing at the genetic level. So there should be no label. The sensitivity should be better than 10 picomolars. The specificity, single point mutation, and you should be able to do that in the complete background of the injected molecules. And if you want to take that a step further, then you have to go beyond PCR amplification, because that is the gold standard so far. So you will see what we have done with this. So the sequencing of the entire human genome about 15 years ago still leaves us actually with a puzzle that we know now, the way what we are made of. But we still don't know actually how we work. And the reason for this is that there is actually less than 23,000 genes are coding for proteins that performs all life function. But that is only 1.5% of the entire DNA, and 98.5% were always actually, to some extent, attributed to junk DNA. But that is not true anymore, because many of those functions are actually happening in that non-coding part of the DNA. And that is seen here, that about 35,000 DNA regions are equal in mammal evolution, and 150 are unique to human. So those are called the human accelerated region, and most of them are actually a part in the non-coding part of the DNA. So people really started to look to alternative splicing, but in particularly into epigenetic. And in order to see this, to find this processes in there. So epigenetic changes can turn on or off gene activity without altering actually the DNA sequence. And the malfunction of these regulatory processes of genes can trigger cancer progression, and the mutation of specific genes disrupt biological pathways, resulting in obviously uncontrolled replication. And this is how nature is doing it. This is called a process of methylation, and basically it. What was that? Oh, not my phone. It happened to me before. Okay. So this methylation process is that it binds actually to a DNA base, the cytosine, and more or less silence the gene. So many maladies are actually polygenetic, and that is a complex interaction between myriads of genes, and it can lead to changes in future generation. So we teamed up at that time with Roche Genetics, and we looked in whether our method is able to distinguish the on and off state of different genes. And we looked particularly into interferon alpha, because interferon is a body-owned defense against cancer. And actually that turned out to be very successful. So what you see here is we plotted here when interferon alpha is present, and here when it is absent. And what you can see here, we used an aldolase gene. This is a housekeeping gene which always is switched on. If it would be switched off, you would sooner or later die. And therefore, references are as I said before, a very important issue here. And you can see that when that's the signal when interferon alpha is present, and if it's switched off, it goes in a sort of idle position. So this work actually was very encouraging for us to see that the method is actually able to distinguish between the state of genes, whether they're switched on or off. And it was the first hint actually for us to see that we presumably had a tool that is applicable in personalized health care. So this work was republished here, and actually to assess patient's condition, what we do, we are using conventional assays, and then we are sort of looking in and look that we can diagnose the relevant biomarkers and the mutations. And according to the Skin Cancer Foundation, the American Skin Cancer Foundation, skin cancer is on the rise. And the reason for this is the sun-seeking behavior of people, and actually it has more incidences than prostate cancer, breast cancer, and lung cancer, etc., per year. And melanoma is its deadliest form. So we teamed up with Donato Rimoldi from the Cancer Research Ludwig Center of the Cancer Research at the University of Lausanne. And we looked to the melanoma, what we can do there, whether we can really show this mutation. So Melanoma comes in different growth phases, and it starts in this radial growth phase, and then it moves into a vertical growth phase, and then it gets really nasty because it contaminates the blood cells and grows metastasis in this way. And this circulating tumor cells then carry this in the bloodstream. So this is the detection of this melanoma-specific mutation in the BRAF. 50% of all the patients in melanoma, they carry a certain mutation, and it's called the mutation is called V600E, and that's part of the sequence there. And here we show basically a concentration dependence of the DNA. And you nicely can see how sensitive we are in the measurements. So this is an injection of the mutations into the cell in the total RNA. Here it's in the DNA. And you can see in this concentration dependence that we actually can measure the wild type and the mutant, this mutant down here. And it takes about 15 minutes in order to get these signals. And it's between about, we are sensitive to about the deflection of 2 to 3 angstroms, 2 to 3 nanometers. And as you can see, it develops, the mutant develops down here to about 80 nanometers. So we have taken this a step further and looked into the total RNA. And I just plotted here these two stages of 100 nanograms per microliter and 20 nanograms per microliter, just to show actually that this takes us down to the lower nanomolar or picomolar sensitivity. Very important, what I mentioned is that this is all done in a single point mutation, as you can see that in the total background. And the references, as you can see here, is just simply an AC mer, and what's very important is that we also can do that against the wild type in the reference. And yeah, so this just shows that, which is very important, that that we can distinguish on the same device. We can distinguish between the mutation and the wild type. So far, this is all done with cell lines. So we decided to make the step forward and take it to the clinics. And we teamed up with Catherine Glatz from the University Hospital, and we have taken the sample from patients, from 9 patients, and which we have been given the sample, that's all. And the analysis was done in a double-blind study, so we didn't know anything. We didn't know anything. So we just got the RNA of these patients. And as you can see here, where they come from is the type of biopsy. For example, here, lung metastasis, and so on and so forth. And this is the RNA, basically what we got, because we are doing it with the RNA because that actually leads to that we can circumvent PCR amplification. So what actually it tells you here is this is the histological status. You see the patient 10102, you see that that is a mutant. We didn't know that. So that came out in our evaluation, and you can see we got 100% right of that. That is just the beginning of this kind of analysis, but it's obviously very encouraging for us to see that atomic force microscopy technology can be moved in. And you can see that there are no false positives or negatives in that. So that is also shown in a different way. This is a hierarchical cluster analysis, which is a dendrogram. And you can see here what happens is when we have the mutant of the cell line and the mutant of the biopsy, it falls absolutely in the correct part here. And the same thing is true, is that the wild-type cell line and the wild-type biopsies also be at in the right place. So that is extremely encouraging for that. There are patients for a more thorough investigation. 10% of people have a different mutation, and this is called V600K. And what we have done here is we have functionalized the mutation V600E and the mutation V600K on the same array. We functionalize that, and then when we inject the complement of the V600E, you can see that reacts very fast. You can see that here just within in 10 minutes. But the V600K doesn't react, and that is what you want here. And then you do it the other way around, and you see that when you inject the complement of the V600K, then that gets into this signal, and the V600E doesn't react. So you actually can toggle with that, and that was also a step forward and a very encouraging result. And as you can see here, again, we do that in a point mutation way. So this just shows that 5% actually of the mutation in the total RNA is enough to get 100% detection. Only 5% of it leads to 100% detection, because this is a kind of important, because this is the COBAS system of Roche that is used at the moment. This is current technology, and this is PCR based here. And as you can see here, we are even better than 5% in order to get 100% detection rate in the mutation. So we are somewhere down here. So again, this is very encouraging. And these protocols, by the way, they can be error-prone with false positives as a particular hazard. And so again, we can circumvent this not using PCR-based assays. So we don't need that anymore. So Roche came out about 4 years ago with the first personalized medical drug that is targeting people carrying this V600E mutation. I have forgot that here. Yeah? That already produced after a year some side effects and also other problems, and therefore No, I show that first, yeah. So this is it's also very expensive. For a patient, for a therapy for one patient can come up to about $200,000, okay? So it's very expensive. This is a 30-year-old man with a BRAF mutation melanoma, and that was the therapeutic effort of the Celboraf, basically. And what you see is that's before the therapy, that's after 15 weeks of therapy, and then the relapse of 23 weeks. And what you can see, actually, the lesions are coming exactly back at the same place. So that's very interesting. But what I actually forgot to say, which is so important for us, and that is, can I go back quickly? I show you this. I have to go back to this. Yeah. Because this is the most important thing. You maybe have spotted it. You know, the paper in Nanoletters of all this work is out today. So I forgot to say that before. Anyhow, it just came into my mind. Anyhow, so this is where we were before. And this is just to show the metastasis. So a new inhibitor in the MEK and in the MAPK signal pathway has been found and converted in a new drug. It's called Kotelic. And this was approved last August, just a year ago. At the beginning, FDA approved it, and it's actually a cocktail. And this is a combination with Celboraf. It leads to a significant extended life expectancy, which means from 6 months to about more than a year. So that is where our current technology is at the moment. So we basically know that we actually can extend that to other tumors, and we took it a step further into breast cancer, and we looked at the human biopsies of people with the HER2 gene, where about 20% of all the patients showed this HER2 overexpression. And this is an electrophoresis quality assessment, and it's very important to see these two RNA bands. So this is how you can monitor this, actually, basically on the histological way, fluorescently in situ hybridization of the HER2, or you extract the RNA again and use our methods. And basically, we just can show that we can first distinguish between ductal carcinoma here and a benign tumor, a malignant tumor, and a benign tumor. And we also can show the comparison between the quality, the different quality. For example, here where these two bands are present, we absolutely can monitor this, or where it is degraded, then we have difficulties to show this. This is ongoing work at the moment, and we've done exactly the same with a double-blind study, and what you can see here is that we have two false positives due to degraded biopsies, RNA samples. This is done from François Huber mainly in our group, and Martina Blodinek, and she is in the pathology department. And those two people are very important because they have carried out all this work. They're actually sitting in the middle of the audience here, and they are part of our team. François Huber is our biologist, and Hans-Peter Lang is a physicist, and completing our team. So the method that I showed you is faster than the standard Sanger or pyro sequencing methods, and comparably sensitive to next generation sequencing. Processing time from biopsy to diagnosis is below 1 day. And we don't need labeling, no PCR amplification, and no sequencing. So that is where we are today. So there is a medical revolution in the making. And this is known as CRISPR and Cas9, where the protein Cas9 can basically be put at any part in the DNA, and then the malfunctioning gene is being cut out and replaced to a donor DNA. This is obviously goes into all species, as you can see here. And just as I'm speaking, actually, the first trial is being done in China and started this with human, started this month. And we think with our technology, we actually could characterize this repairs. So that's why I'm showing this. But this is absolutely revolutionizing new technology. Is accurate, low cost, and easy to do. So that is the future. There's no doubt about it. So going back from the small genes and mutations up to the cell, and so we want to actually follow whether we can capture the CTC, the circulating tumor cells, and melanoma excels cell expressing high molecular weight melanoma-associated antigen. This is what you can see here. And then we sort of we functionalize the device and use the antibodies covalently attached to the surface. And obviously, because that is a very important thing. The number of the CTC is very low. So it's about 1 to 10 in a billion cell per milliliter blood. So they're extremely scarce to be detected. But we gave it a try. And that is what it looks like. Go back. Okay, so again, we functionalized only four of the sensors are functionalized, and we use the others as the present thing. And what you see here, of course, immediately is the differential measurements actually shows much bigger deflection of the whole sensing. That is now in the micrometers. It's obviously because you have the cell attached to the device. And again, this is the reference, and the reference should not give any signals. So this is the way we've done it, and this is actually the real-time capturing of this CTCs, and that is a mixture of melanoma cells and lymphoblast cells together. And when you see this, that now it works. So as you can see, it seems that all of the arrays are functionalized, but that's not true, because what you see down here is they only bind. Gerd said that, it's the binding, what binds to what. And so here, this is those 4 senses are where antibodies and antigenes are binding, and actually there are. We are very confident that we can take that further. It's about 10 cells at the moment, the sensitivity. So what we are doing actually is combining the top-down and approach and the bottom-up approach of self-assembling, self-organization here, and which it's so wonderfully implemented in the natural world. And when we actually started to do this, that was together with way back, together with Jim Jimjewski. He is also here. And we never could foresee at the time that we at one day would actually get into the clinic, basically from the bench to the bedside. So George Gamow stated that took less than an hour to make atoms, a couple of 100 million years to make galaxies, stars, and planets, but more than 4.5 billion years to make a human. And life indeed is complex and very, very complicated. So I'm going to show some of the state-of-the-art work that has been developed in recent years with AFM. So I'm going back to imaging now. Okay. So Toshio Ando at the University of Kanazawa developed the high-speed AFM, and so that you can actually look at molecular interactions in real time. That is. And you can see that you can see this here, you know, this, for example, this translocation along an actic film with this myosin molecule, or the molecular motors, that is actually shown here. I don't know why it doesn't work. No, it works. Okay, and this is a new development that actually takes us, with this high-speed AFM, it takes us in the time domain of chemical activity, and certainly also you see the power of high-speed AFM monitoring the cellular machinery at the nanoscale and in millisecond resolution. So this has been developed by Toshio Ando and his colleagues in the group, and it took him 15 years to get there. I mean, it's a work of 15 years, and you can imagine, you know, nowadays when you put forward the proposal, nobody would give you any money to do that, you know. So it's quite miraculous that this is happening. So, and this is certainly a way for application. So the nuclear pore complex is part of the nuclear envelope, which you see here. This is the cell, and this is the nucleus. This is the nucleus. Pore complex is part of the nuclear envelope, and it's a selective barrier that controls the transport of molecules between the cytosol, that is out here, and the nucleus. And there are a few models actually came forward. This is just published in this month, Nature Nanotechnology, and there are 3 models. And actually, what I'm going to show you is a compelling evidence that this is the right model. And what Gerd already said, that people could already imaging similar molecules, but here you actually see it dynamically. So you bring dynamic in to the whole situation, and you see this nuclear complex in action. And you can see that where just cargo is transported into the molecule. So this is a work from Rod Lim and Yusuke Sakiyama at our institute. And then Gerd already showed, no, this is in between in biology. So what happens when cells are going through mitosis, and they're going through a rounding, and you just can basically measure the forces that have been created during that process, and with a cantilever that measuring the forces. You also can do the opposite. You just could hold on to it, and that would lead to a cell lysis, you know. So, and you could see that here, this is the chromosomes have been fluorescently tagged. And what we have done now together, this is work from Daniel Miller's group at the BSSE ETH Institute in Basel, and we have now developed a completely new AFM that actually is enabled us, or will enable us to follow and measuring the masses during mitosis and future generation and daughter cells. So Gerd said something about this. This is, again, work from Leo Gross, and you can basically discriminate between chemical bonds in this hydrocarbon arrangement of 13 fused benzene rings. And you can actually look at the correlation of the bond order between the central and the outer ring. And the length difference is 0.003 nanometers. So that's as good as it gets. So this is new work from Ernst Mayer's group. He's sitting in the audience, and what you see here is taking care of friction. Gerd mentioned that before. So when you do that with this nanoribbon on gold, this is a nanoribbon here. This is a nanoribbon on gold. This is just one atomic layer. This is a graphene nanoribbon, and basically if you drag that along the gold surface here, then the friction is absolutely vanishing and gets into a state of super lubricity. So this is Shigeki Kawai and his group who does that. That's just also, you see how dynamic the field still is. That is that was just published a month ago. And in the same group, that just shows the high resolution of the AFM with its CO tip. So what Gerd said, that you pick up this CO tip, this is actually part of Pauli repulsion, that you get this high resolution. This is the STM, and if you look at the AFM picture, then you get the absolute resolution, and you see that that AFM has actually surpassed the STM in many ways. That is documented nicely here. So sculpting was mentioned. So what you see here is a sodium chloride island on a copper surface. And sculpting, you can actually use the cantilever as a chisel, and then you sculpt. For example, you can do that here. And what happened here, this is about 200 atoms, and this was actually done in order to follow the contours of Switzerland, and this presumably the smallest map that we know, in Switzerland anyhow, I don't know otherwise. Anyhow, so if you have a gifted person like Shigeki Kawai, he can then, at room temperature, molecules, he can then manipulate this bromine atom and arrange it in a Swiss cross. So this is actually work from Ernst Mayer's group at our institute. Okay. This is how it all started. This is how it all started. And it has some, that's the first AFM, and it has some analogy with this. Okay, that's the first transistor. But what it actually tells us, what it actually tells us, for a proof of principle, not necessarily you have to build a Ferrari. Okay, that's not for a proof of principle. That tells us. However, the sole idea actually to develop the AFM was to get atomic resolution on non-conductive surfaces. But people obviously have picked on that and developed it into presumably the most versatile and most powerful surface characterization tool that we have today. And this is testified by more than 350,000 papers published in peer-reviewed journals. And that's where we actually are today. So the first AFM lives in the Science Museum in an exhibition called The Making of the Modern World. And I believe the Apollo capsule behind it is in good company. And the reason for this is because an AFM has been already on Martian soil, and one is on the way, or on this European Rosetta mission to this Chury comet. And actually, it seems that not even the sky is the limit for this technology. But we are not yet stardust, the three of us. So this is the only picture. This is the only picture of us after we developed the AFM until yesterday. When that was taken by Jim Jeffsky. He is here, and that was actually at the occasion when the three of us met again at IBM, and that was taken in a nearby restaurant. So with that, I conclude my talk and just tell you who we are. Just a small group, interdisciplinary. Everybody has a different background. People who help us, and those are people who we work together from other institutes. Thank you for listening.